Secondly, because the substrate molecules possess more energy, the likelihood that the reaction will actually occur increases, because the greater proportion of molecules have energy that is equal to or greater than the activation energy, and so can actually react when they collide. As you saw with pH, the graph reaches a peak at the optimum temperature for the enzyme, and then it begins to decrease. At high temperatures, the structure of the enzyme begins to break down, because the molecule is moving so actively that the weak bonds which hold the protein structure together in its normal three-dimensional shape become strained and eventually break, so that the protein becomes denatured. In the same way as the pH optima, different enzymes have very different optimum temperatures. Within animals, enzymes work best at about 37 degrees, which is normal body temperature. However, enzymes from organisms which have evolved to survive in extreme conditions can have very different optimum temperatures. The DNA polymerase used in PCR reactions to replicate samples of DNA comes from an organism called Thermus aquaticus, which lives in hot springs. Now, this enzyme operates best at a temperature around 75 degrees centigrade, a temperature which would immediately denature proteins from animals or humans. Now, we're going to look at a different enzyme called lactate dehydrogenase. This enzyme is very important in muscle tissue, where it converts pyruvate to lactate in anaerobic respiration. Here is the reaction that it catalyzes. You can see that it is reversible. You can see here that during the reaction, NADH is oxidized to NAD in a 1-to-1 ratio. So for every molecule of pyruvate that's converted to lactate, 1 molecule of NADH is converted to NAD. NADH is the cofactor for this enzyme. It's needed to accept the hydrogen atoms that are removed from the pyruvate when it's converted to lactate. This is really useful in terms of being able to monitor the activity of this enzyme, because NADH absorbs light, but NAD does not. So we can use the disappearance of NADH, and so the increase in absorbance, to follow this reaction. Here is a solution of NADH. You may be surprised that it isn't colored, since I've just told you that it absorbs light. However, in this case, NADH absorbs light in the ultraviolet range at 340 nanometers, so it doesn't look colored to our eyes. Once again, I've set the spectrophotometer to 0 using a cuvette containing the components of the reaction which we don't want to monitor. And I've set up a cuvette containing buffer at a suitable pH, the substrates for the reaction, which are pyruvate and NADH. When I put this into the spectrophotometer, you can see the absorbance caused by the NADH present. And I can monitor the cuvette for a few minutes to make sure that the reading is stable and that there is no reaction in the absence of the enzyme. Now I add the enzyme. This time we're using a continuous assay. We're monitoring the reaction as it progresses. And you can see the absorbance value decreasing over time and being recorded on the chart. You can see that to begin with, the reaction rate is constant, but then it begins to slow down. This happens for two reasons. Firstly, the concentration of substrate available decreases as it's used up. Secondly, you will remember that this reaction is reversible. So as the amount of product increases, the rate of the reverse reaction also increases. Eventually, we reach a point where the absorbance stopped changing, not because the enzyme has stopped working, but because the reaction has reached equilibrium. Now, the forward and backward reactions are working at the same rate, and so you don't see any overall change in the amount of NADH or NAD present. Using the printout, I can now look at the rate of decrease in absorbance and use that to determine the reaction rate per minute. To do this, I draw a line through the linear part of the graph and measure how much the absorbance changes over a 1-minute period. If I repeat this reaction at a lower pyruvate concentration, you can see immediately that the slope of the line is shallower. The rate of reaction is slower. If we plot a graph of reaction rate against substrate concentration, you can see that as the concentration of substrate rises, so does the rate of reaction. The explanation for this is based on collision theory. The more substrate molecules there are present in the solution, the greater the chance of a collision taking place between a substrate molecule and an enzyme molecule. So more reactions take place in any given time. However, as substrate concentrations continue to increase, the increase in rate levels off because the enzyme is coming close to working at capacity. It's catalyzing reactions as fast as it can. So at low substrate concentrations, it's the availability of substrate that limits the reaction rate. Whereas at high substrate concentrations, it's the concentration of enzyme that's present that is the limiting factor, i.e. the number of enzyme molecules present capable of catalyzing a reaction. The experiments I've shown you today are examples of the type of experiments that you might conduct during your undergraduate course on degrees in biochemistry or other biological sciences subjects. Enzymes are a very important area for research. Understanding how they function allows us to understand how cells and tissues work. Inhibiting enzyme reactions can be an important part of treating common diseases, such as arthritis or AIDS. Being able to monitor enzyme activity is a vital tool in biological research.